It's my pleasure to welcome you to this year's G. Burroughs-Mider lecture. Our lecturer today is Wei Yang, who is a senior investigator at NIDDK. The lecture series honors the memory of G. Burroughs-Mider. I would guess most of you in the audience don't remember who he is, but he was the NIH director of laboratories and clinics from 1960 to 1968 under the leadership of James Shannon, a legendary NIH director. And during his leadership, there was an enormous expansion in the size of the intramural research program. And during those glory days in the 1960s, there were 4 Nobel laureates working at the NIH at the same time. So it was an amazing period. The lecture in his name recognizes an intramural scientist whose work exemplifies the creativity, fearlessness, and excellence that characterizes research in the intramural research program. The speaker is chosen by the NIH director from among many nominations given to us by intramural scientists and intramural leadership, which are vetted by the board of scientific directors and then move forward for the director to consider. This year's Mider lecturer is Wei Yang, a senior investigator and deputy chief in the Laboratory of Molecular Biology in NIDDK. She's used very creative structural approaches to revolutionize our understanding of the interaction of DNA with proteins during replication, repair, and recombination. Dr. Yang received her BA in biochemistry at Stony Brook, her PhD in biochemistry and molecular biophysics with Wayne Hendrickson at Columbia, and postdoced with Tom Stites at Yale. She's been in the Laboratory of Molecular Biology in NIDDK since her arrival at the NIH. Dr. Yang has appreciated the need for detailed crystallographic studies of the way in which DNA and enzymes involved in replication, repair, and recombination interact. This work has provided new insights into the function of the DNA repair proteins MutL and MutS, the first co-crystal structures of Y-family DNA polymerases bound to DNA substrates, VDJ site-specific recombination complexes, and the structure of RNase H bound to RNA. Dr. Yang's work is characterized by a combination of elegant biochemistry and high-resolution crystal structures to gain new understanding of biological function. Her talks, as you will see, and those of you who've been fortunate to hear her talks before will understand this, are models of very clear communication and use aesthetically beautiful structures to clarify new biological concepts. Dr. Yang's work was recently recognized by the Dorothy Crowfoot Hodgkin Award, a high honor indeed. Her talk today is entitled Genomic Integrity and Cancer Prevention, Molecular Mechanisms of DNA Repair. Wei.
Thank you, Mike. Thank you for introducing me. I was very surprised when Dr. Michael Gottesman told me about the news that I was selected to give this year's Mider lecture. As one of my professors said many years ago, that the best word we scientists work for is peer recognition. And I feel very honored and humbled to receive such distinction by my peers at NIH and the directors. And when I preparing this lecture, I couldn't stop asking myself, what I have done truly matters to biomedical research. And a couple of days ago, I was alerted by Mike Lichten that my talk announced on NIH website is 3 a.m., February 22nd. And it dawned on me that what really matters is that we are willing to work very hard at any time to make a difference. So what I'm going to tell you about today is something related to DNA replication. And DNA replication is essential for cell proliferation. For every cycle of cell to divide, before that genomic DNA have to be replicated. And the cell really make a big decision using a lot of checkpoint before going into the replication cell division. But once it's getting, it has to complete in order to survive. If the replication is incomplete, then cell dies. So it's critical. And associated with DNA replication, we study two events. One is translesion DNA synthesis, one is mismatch repair. And translesion synthesis is necessary because not all DNA is perfect. We go out today, exposed to sun, the DNA can be, the adjacent bases can be cross-linked. If we eat too much barbecue food, we may have ingest some carcinogens and DNA base gets damaged. And those need special DNA polymerase to pass through because they blocked normal DNA polymerase. On the other side of the coin, when DNA is made by DNA polymerase, polymerase is known for very high fidelity and make very low errors. But in a genome of 3 billion base pairs, even the error made in the ratio of 1 in 10 million base copied, we're still going to have tens to hundreds of misinserted base left by the DNA polymerase. And that could be base are paired wrongly or base have no partners. And those are taken care by mismatch repair. And to bypass the DNA blocking, replication blocking lesion, and to repair the errors made by DNA polymerase are just two sides of the same coin. The purpose is to keep the genome have minimal mutations. As everyone knows, cancer rises because mutations, and those two pathways directly implicate on reducing mutation rate and the cancer incidence. And I want to start my talk telling you what we do. And in our lab, we use X-ray crystallography as the main technique. What we do is starting from a gene that's been identified that's causing human disease. And if we are lucky, we have very wonderful people. They have just discovered some new pathway and new protein. Before they publish, they approach us, structural biologists. Then we get started. And I have two very good colleagues start me on the translesion synthesis and mismatch repair at NIH. That's Roger Woodgate and Peggy C. And once we identify the right protein, then we generate enough protein to make crystals, and the crystals may be beautiful, maybe not so pretty, but the important thing is that they can diffract X-ray, and we can record those spots on film or on various recording device nowadays is very advanced. And then using those information, we can generate electron density maps, so-called, and then build a structure model. And then use the model, we start to interpret what the model means and what the structure tells us how the molecule works. And with advanced technology, growing crystal become easier. We have robots shown here, funded by ITAP, and we have synchrotron radiation source in Chicago. We don't even need to go there. Tonight we're going to collect data at home in office, and we just ship the crystal by FedEx. And so we spend more and more time to understand the function, because structure is pretty, but it doesn't speak to us. It doesn't tell us. Very rarely when you solve a structure, you immediately can understand how it works. So we spend a lot of time trying to understand that using biochemical cell-based tools. And the other thing it's the advanced technology allows us to do is instead of study one single small protein, now we can study large protein complexes made of multiple proteins, a macromolecular assembly, because no protein works singular alone in cell. So the first part I'm going to focus on is called mismatch repair. That's repair the mismatch incorporated by DNA polymerase. Here's just a story from this family. It's heritable, and 1 in 20 colon cancer patients identified diagnosed in America carry the mutations that's deficient in mismatch repair. And according to the American Cancer Society, about 1 million Americans carry one of those defective mismatch repair genes. And those people have 80% of a chance to develop cancer in their lifetime. So it affects many of us. And the 4 genes encoding 2 special proteins called the MutS and the MutL. And we started a project in bacteria because it's easier to handle. And the MutS and the MutL are conserved from bacteria to human. And here's demonstration of black line is template DNA, the red line is newly synthesized daughter strand. Unfortunately, there's a misinserted base by the polymerase. And the MutS is the protein universally recognizes this single mismatch. And then it recruits MutL and the nuclease to make a nick in the daughter strand. And in E. coli, daughter strand is transiently unmethylated, while template strand is methylated. So the nucleus will make an incision in the unmethylated daughter strand at the GATC site. And once nick is made, the MutS and MutL together recruits the helicase to unwind the DNA from the nick, because the GATC sequence can be anywhere in the genome, and usually the hundreds base pair away from the where mismatches. The UvRD will unwind the DNA, and exonuclease will remove the daughter strand, and then afterwards, the whole strand will be resynthesized in that patch of hundreds nucleotide. As you can see, the process is extremely expensive. Remove hundreds of newly synthesized the daughter strand and resynthesize again. But this process is conserved from the human as well from E. coli, and the MutS and the MutL is playing an essential same role, and exonuclease is required. In addition, the process actually contains 3 ATPase. In E. coli, MutS and the MutL, UvRD are all ATPases. In human, at least MutS and the MutL are ATPase. UvRD is not conserved in human. Interestingly, MutS is an ABC ATPase, which is known for ABC transporter. MutL is so-called GHL, a different ATPase. And the UvRD is a bona fide motor protein. And the people here maybe think ATP can do many different things, but most people will, you ask them what you think about ATPase, people say, oh, it's a motor protein. But what I'm going to show you here is that MutS and MutL are not just using ATP as a motor, actually doing something different. We call it a thinker because it will have to make a decision whether MutS really recognize a mismatch. And the MutL, we think it's a talker. It recruits protein to come to the site to do the repair. Only the UvRD is a true ATPase motor protein. And I'll start from the simplest ATPase because that's the function everyone understands. Motor protein, what motor protein does is burning ATP. What it does is the energy is used to organize the motion from non-directional Brownian motion to directional walking. But energy alone isn't enough to make directional movement. Why so I say so? Because if you drop a person into a virgin forest without any path, the person can walk in the forest for days in Brownian motion, never get out because it doesn't know where to walk to, unless he has a GPS. But if there's a path, then one can walk on the path and get out of the forest. And the motor proteins are just like human. In order for them to walk in certain direction, they need a track. So everyone knows myosin walking on actin filament. The actin filament is the track, and the myosin is the motor. And for us, we really want to understand how it walks. But having both motor and the track, it's way too large and too heterogeneous for crystallography to do. And showing here is a picture recorded using AFM, atomic force microscopy, and a video recorded by the group of Ando in Japan. And that's probably the state of art right now. You can watch a motor move. But you can see it's very hard to really see the detail. And then one myosin walk quickly and the one gets stuck. You don't know what happens there. And it's just like you using a watch. If the watch is telling you the perfect time, the needles are moving, you're satisfied. But if the watch is telling you wrong time or it doesn't work, then you need to look a little closer. To open the watch, look at the detail. And to really understand how motor protein works, we really need to have a closer look. However, tempo, the time resolution and the spatial resolution really is going against one another. If you want to see the real time, you have to drop the resolution. If you want to see the high resolution like X-ray crystallography, we never can record a dynamic process because we only crystallize one conformation at a time. But that sort of incompatibility can be resolved. Using the example by Edward Maybridge, probably many people have seen this movie. It looks beautiful, and it was recorded way before there was video camera. It was done in 1878. And the way he made this movie, the frame per second is fewer, but he was able to make it. The way he made it is by strategically placing camera around the racetrack. And I still don't know if those pictures taken sequentially or he sorted the picture and then spliced them together to make a stop-action movie. But that gives us an idea that crystallography can use the same way to make movies. We may not be able to make like 13, 12 panels of the picture, but we maybe make 2 or 3 very essential stages of the molecule, how that works, and then splice them together to make a movie. And that's exactly what we did. So we crystallized UvRD with DNA at one critical state. Then we crystallized the helicase with DNA and with a non-hydrolyzable ATP, the energy source. And again, we crystallized DNA with helicase. Now we co-crystallize with ADP, which is a hydrolysis product of ATP. And with this static image, you can already see the DNA leaning angle is different. If you count the base pair from this point to that point, they're different between binding ATP versus not binding ATP. But I will make your life a little easier by stringing it together as a movie. And the work was done really thoroughly by a postdoc, Jae-young Lee. He solved many structures to identify those 3 states. Now you can watch. ATP floated in. It comes in and induces a motion between this portion of the protein to that portion of protein. It's a rotation. And coupled with that, the base gets unpaired because the DNA in a screw motion comes down and push against this hairpin, and the base closest to the hairpin gets separated. That's just during ATP binding. And in the ADP-PI release motion, you see the translocation of single strand occurred. So we really break down the ATPase cycle into 2 fronts. ATP binding induced the unscrewing of the double strand, and the one base gets unpaired. And after ATP hydrolysis, ADP and the PI release allows the single strand translocation to complete the cycle. By making this 3-state structure observation and put them together, we can answer with confidence that one ATP hydrolysis allows one base pair unpaired. And we know exactly where the protein-DNA interaction occurred. And of course, at some day, we'll be able to use it to answer, to really help the human understand how there is helicase in human body work. And the work we did here actually is up to date. That probably was finished in 2006, but probably is still the best structure description of how a helicase work to now. And now I will switch gear to the second ATPase, which is MutL. And it's a dimer, and it's conserved from bacteria to human. The most conserved portion is in the N-terminal, have 350 residues, very conserved. And of course, we said, well, let's crystallize it and see how that works. And the postdoc, he was my very first postdoc, did the work. And he solved the structure. And actually, after structure solved, we were quite disappointed. The reason is that out of 350 residues that's crystallized, we only can see 280. 70 residues were disordered. Among those, many are very conserved. So we had to leave the structure on shelf for a while, try to understand what the structure means. And while we were busy doing biochemistry, we stumbled on that this protein actually is an ATPase, although in the literature it was labeled as has no enzyme activity, and we have difficulty to publish the paper because it's known to have no enzymatic activity. But at the same time, when we discovered it can hydrolyze ATP, there were 2 papers from bioinformatics study group, one from NIH, Eugene Koonen's lab, identified that MutL actually bears sequence homology to a very well-known ATPase, that's gyrase. And the DNA gyrase actually was discovered and characterized on this campus by my colleagues of Kiyoshi Mizuchi, who is sitting here, Marty Gellert, and Howard Nash. So then it's all speaking that it's an ATPase. So we went ahead and recrystallized the protein in the presence of ATP. Now the structure looks very different from without ATP. The one shown in orange are those 50, 60 residues now upon ATP binding become ordered. And not only become ordered, some of those newly formed secondary structure formed the interface for the protein to dimerize. And this scheme of ATP-induced large conformational change actually is applicable to the whole family of this contained gyrase and also heat shock protein 90. And so I'm just recapitulate this movement of ATP-induced morphological change in this movie, showing you again the protein starting looking pretty pathetic. We call it ugly duck because it's missing a lot of things. Once ATP bound, you see it induced a huge amount of conformational change, secondary structure formation, and the protein goes from monomer to dimer. And the reason that it was very difficult to identify that this is actually an ATPase, heat shock protein 90 was not known for ATPase, then thought was ATPase, was denied as an ATPase for years. The reason that the ATP turnover rate is extremely slow. MutL actually only turn over ATP 1 per 5 minutes. And you kind of like, what does it do in 5 minutes? It turns over so slowly. And we kind of was looking for what can control the speed of ATP hydrolysis. What we find is that we made a bunch of mutations along the pathway. We can block it from ATP binding, block from dimerization, block ATP hydrolysis. And we delineate that when the MutL bind to ATP, actually that's the only state it can activate in the nuclease. And once the nuclease make an incision, then DNA have single strand exposed, and the single strand DNA will speed up this portion and reduce time dwelling here and increase time in the free form. And the whole protein, both the N-terminal ATPase and the C-terminal dimerization and the whole cycle of ATP hydrolysis slowly by MutL is essential for it to activating UvD helicase. And I told you UvD helicase can unwind DNA, but by its own, it doesn't randomly go onto DNA and unwind duplex. It needs a signal from mismatch repair pathway and need to be held hands to do it in hundreds base pairs. And here's just a model we think how that works. UvD is turning over ATP quickly and unwinding hundreds base pairs per minute. And the MutL is holding it together on DNA, and the MutL itself turning over once in a few minutes. So it's only when the DNA become tangled because so many hundred base pairs unwound, then MutL let it go and reactivate it if it's needed again. So those two stories of ATPase is relatively still straightforward, how the ATP changes structure or how ATP serves as an energy source to allow motor to work. Now I'm going to the most intriguing ATPase of the family, that's the ABC family, MutS. And the MutS role is recognize DNA mismatch. There are two types of errors that DNA polymerase make. One is just misinserting a base against the template, listed here. But in human, for example, we have a lot of repetitive sequence known as microsatellite, and the polymerase may skip forward or backward, and the resulting unpaired loops called insertion deletion loop in DNA. So basically those have no partner in the opposite strand. And the mismatch in this can happen on either strand. The MutS doesn't care, but it needs to find them. And the ratio of the occurrence, frequency of the occurrence of the mismatch is about 1 in 10 million base copied. So you think about it. MutS protein have to search through the whole genome, and once finding it once in 10 million base pairs. And to find a mismatch, which don't look very different from normal base pair. It's just like, GT base pair, it's not GC base pair. And it's a tremendous task that how to find this one mismatch buried in tens of millions of normal base pairs. So it's very reasonable to liken it as finding a needle in a haystack. So that's very intriguing, and all the mismatch have different shapes. And the Peggy C approached me before she published a paper of cloning MutS, and it's wonderful protein, it's heat resistant, and her postdoc even better. Galia, actually, while we were sidetracked by some crystal that's not diffracting well, Galia actually grew the crystal of MutS complex with DNA, and we solved the structure, and we were the first one actually solved ever MutS of DNA complex together, and here's the movie. And the MutS is a fairly large protein, have two subunits, green and the blue will be always the one will be showing that's interacting with the mismatch. Because even though it's a homodimer, in sequence they're same. In bacteria, the structure is different because there's only one DNA strand that contains the mismatch. The blue subunit is interacting with the mismatch here. But each subunit have two domains, sort of one on top, one below, holding the DNA together. And the DNA is quite bent. And we determined multiple different mismatches with TAC MutS, and a few months after, but we managed to publish together Titya 6-mer in Holland solved E. coli MutS bind with this type of mismatch. And I'm not showing you a different structure because they look just the same. And some 7 years later, Lorena Beasy at Duke solved human MutS alpha, human protein that caused colon cancer, and again look very similar. And in human, the blue and the green are sequence related, but not exactly the same. And the blue subunit again recognized the mismatch. And after solving the structure, of course we have cancer implication, but before I tell you that, I'll tell you a little bit of molecular mechanism. The thing really bothered us is that how MutS really find the mismatch. One thing in common is that DNA is bent. And the second thing is that how does it increase the specificity? Because the difference for MutS to bind a normal DNA versus a mismatched DNA, it's only 100-fold. It's not enough to explain finding a mismatch in 10,000s of normal base pairs. Turns out that MutS is an ABC ATPase family and have the ATPase up there. And you would think that maybe adding ATP, then the protein will bind to the mismatch more tightly. But exactly opposite. Adding ATP, MutS falls off on the mismatch. So there was all kind of proposal that MutS is a motor protein because it's ATPase, and it moves away from mismatch to finding nucleus cleavage site. Or it doesn't hydrolyze ATP and moving as a sliding clamp. But seeing the structure that DNA is so much bent, we just don't think it can slide so easily. And also, if it leaves mismatch, what will happen to the mismatch? It's not even repaired. Is there going to be a second MutS coming and there's going to be a huge mess there? And why shouldn't it stay? So a postdoc, Murray Junep, designed this experiment to test whether MutS needs to leave mismatch in order to activating the repair process. And so normally the DNA are connected. There's a mismatch that's the nucleus cleavage site. Here's the MutS with ATPase binding there. All he did is he cleaved the DNA in the middle. And if the protein MutS have to travel along DNA, hydrolyzing ATP or non-hydrolyzing ATP to come to the site to activating nuclease. When DNA is cleaved, the enzyme should be unable to activating mismatch repair. And as a result is that after DNA is cleaved, MutS still can activating the nuclease, and it requires MutL to bridge it. So it suggests to us is the protein-protein interaction is important. It's not the traveling along DNA. And later on, John Hayes' lab did a different experiment in using human protein. He blocked the DNA path using biotin and streptavidin and show again that MutS can activate a nuclease even in the presence of a blocker. And the kinetics of activation is the same. So based on that and borrowing the idea from that high energy cofactor used in protein synthesis by ribosome, a G protein can use GTP to increase the specificity of amino acyl tRNA synthesis, selection. So the idea actually came out when I was talking to Kyush about this problem. So we proposed this model that MutS binds to normal DNA and ATP helps it to release from the normal DNA so that it has a chance to get on mismatch. On mismatch, once it's there, when it's both bound to mismatch and ATP, it can recruit MutL. And the two proteins together now, even in the presence of ATP, can bind the DNA mismatch stably. And this data is generated by many different labs. And the model we propose is still in the testing stage, and we still cannot exactly tell why it binds to mismatch. The ATP hydrolysis is different. But when the two together, it's been shown by many groups that it can activating the nuclease, and as well as by us, and activating the helicase. And so that was a puzzle, and it's lasted for more than 10 years. Recently, Shikha Gupta, who was a postdoc in Marty's lab and worked with me as well, she solved the structure of human MutS beta. And the MutS beta recognized DNA that is not a simple mispair, but have large base loops that's having no partner. And Shikha worked very diligently for more than 4 years and solved the MutS complex with DNA contain 2, 3, 4, and 6 unpaired base, and have 4 different crystal structures. The reason we solve many structures, sometimes crystal structure are not exactly is unbiased because crystal lattice interaction. And having 4 structure, we really have a very complete view. Here is the original structure of bacteria and human MutS alpha that recognize mispair or single unpaired base. And the Shiga structure look like this. On first view, it doesn't look all that different except to have the top like a corkscrew. Now it's tighter twined between the green and the blue subunit. And in fact, this portion actually is conserved in all MutS protein, except when we were studying bacteria protein, we removed it for easy crystallization. And in the human MutS alpha structure, this just gets disordered in the crystal, so never seen. But the real interest difference I will show you in the other slide when we zoom in, here doesn't really show. So Shiga have different number of base unpaired in the loop. So we are showing you the 2 and the 4 and the 6. What you see is when the loop size changes, the DNA bending angle is very different. And the bending angle is proportional to the loop size. And all those different sizes of loop are recognized and repaired. So that exactly the DNA shape may be not important. How much it bends is not important. And the second thing we learn from here is that compare the bacterial MutS and the human MutS beta, even give just 2 base pair unpaired, same DNA substrate, the unpaired base is flipped out in opposite directions when it's complex with bacterial MutS versus human MutS beta. So exactly where the mismatch locates relative to the protein is not even important because they're all repaired by different MutS. So what does it recognize? And clearly that one common feature you see is that the DNA is bent. It's just bent by different angles. That comes to an analogy. Maybe a good DNA is just a good spring, perfect, and containing a lesion, which is true for any DNA lesion. Whenever there's a lesion, the spring is no longer as rigid. It becomes more bendable. And so how do you tell the spring is perfect, that you can stretch, it will go back, versus spring, if you stretch, it's broken, it won't go back? The best way is jumping on it, right? Give it some energy, and to see if it can resist and it can bounce back. And so that's what we think what MutS is doing. Once we know what to look for, we find the difference. There are 2 subunits. On the right, now it's multicolor, but it represents the blue subunit that recognize that DNA is containing a mismatch. On the left is the green subunit. And the difference is in the DNA binding domain. Now you see that the one that binds the mismatch, this domain, the blue domain, is way down. And this subunit is not involving direct reading of mismatch. It's way up. It's like a crane. One leg is straight, standing up. The other leg is bent. The standing one is looking for somewhere DNA can be bent, and it can insert this domain and straighten down. And the other one is flexible. And what's really interesting is this subunit is present in both human MutS alpha and MutS beta. And the 2 structures have very different conformation of this little blue domain. It's basically free to move without directly interacting with the mismatch. And by able to move like this, this affects how it's hydrolyzed ATP. This subunit binds ATP and it's occupied by an ADP in the crystal structure. And this subunit, because this domain is pulled down, the ABC family ATPase is occluded for ATP binding. So that leads to the explanation why ATPase hydrolysis kinetics is different. That was a data observed by 3 different groups. And we were one of them. We never published it because we were just behind. But it's always sort of a puzzle that MutS will hydrolyze ATP very well with a birth phase, and this is time, this ATP concentration. And with normal DNA, it hydrolyzes this. But if you present a mismatched DNA, the ATP hydrolysis birth phase is eliminated, meaning that the hydrolysis, any step before hydrolysis is very slow, and this sort of slowdowns because product inhibition. We didn't understand this for a long time. Now suddenly we have an answer. That is, when it's binding to the normal DNA, the DNA is like a good spring. It's resist of being bent. And this 2 domain is trying to bounce on it. and DNA wouldn't budge. It's just sliding along, and the 2 ATP hydrolysis goes on very strongly. But once it binds to a mismatch, the blue subunit, the DNA binding domain gets pulled down, and ATP hydrolysis gets inhibited. It cannot bind ATP very well. And the green subunit still can bounce up and down, but the overall ATP hydrolysis is much slower. And what does it mean? It means that we have a model how MutS identify mismatch, because using the ATP as a proofreader, it binds normal DNA, it tried to bend it, and the DNA is very strong, resists to the bending, and allow the ATP hydrolysis to going on very strongly. And so the protein dissociates from the normal DNA or sliding along DNA. And if it reaches the mismatch either by direct association or by sliding, because the mismatch, the DNA can easily be bent, and the blue subunit find an easy footing. And once it gets stuck on this bent DNA, the ATPase gets inhibited. And then the blue subunit hydrolysis alone of ATP is not enough to dissociate it, at least for a time duration. And by green subunit capable of hydrolyzing ATP and the blue subunit binding to the mismatch, the two together, then they recruit the next protein for forming a repair complex. So, and we are still doing experiment, and a lot of people out in the world will probably also doing experiment to either to prove or disprove it, but so far it's satisfying, agreeing with the kinetic data and the structural data. And then now I'm finally going to return to the point when we started mismatch repair, the cancer, how it's related to colon cancer and how we're going to helping, knowing all those molecular mechanisms to help understand the cancer mechanism. So it was really nice to have the whole pool of data, hundreds, thousands data points that mutations found in the cancer patient. So we can, once we have structure, we can project them where they are. And wherever there is causing cancer, then you say, well, that's functionally important. But it wasn't that we always can zoom into the right place. When we first found it, immediately it's noticed that the ATPase site is important because a lot of mutations cause cancer. And then this long alpha helix link from the ATPase to DNA binding also caught our attention. But it's only until very recently when we noticed this bouncing effect of the DNA binding domain, we appreciate that this interdomain connection interface are so important for communicating the ATP activity to DNA binding and allow the ATP energy to use to test whether the DNA is containing a mismatch or not. And furthermore, we know that now that between the communication between two subunits, the interface up there is important, and something that's exposed to solvent, we don't know why, and we can only propose maybe it's important for binding to the MutL or other factors in mismatch repair. So that's how we use the cancer data in terms of helping. It's not a good drug target because it's malfunctioning, that's why we have cancer. But what the structure understanding of the molecular mechanism is that now if we find a mutation, we can help to identify whether the mutation actually is truly causing cancer or maybe it's just a polymorphism and how serious it is. And so it really helps diagnosing. And we are a long way away from why certain mutations in certain patients cause colon cancer. In women, for example, it's not the colon cancer, it's endometrium, ovarian cancer, or brain cancer. So there's a long way to go, but understanding molecular mechanism certainly empowers us to fight war against the cancer. And I'll switch gear now for the next 10 or 15 minutes on the DNA polymerase of translesion synthesis. I introduced before that DNA are not always perfect. You need special polymerase to bypass those roadblocks. And years ago, when Roger Woodgate found a very nice DNA polymerase, which represents a new family in archaeo, he approached me, and we together solved the structure, and Honlin, the former postdoc, solved the structure. And here just showing you a good polymerase that makes very few errors, and the polymerase that makes many errors. And the difference is in the active site. Here you can see the active site represented by two blue spheres. That's the metal ion required for catalysis. Here you cannot see it because the active site for the good polymerase is hidden inside, and it's much more enclosed and very much more strongly discriminating against the wrong base or not good template. But this is not exactly telling us how human works. In human, actually, there's a homolog of this low fidelity DNA polymerase, and people who have mutations in that genes have high sensitivity to UV light. And normally you know that we go out to enjoy the sun because it helps us to strengthen the bone, converting cholesterol to vitamin D. But those people who have deficiency to repair UV lesion, they're very sensitive and they have very high incidence of skin cancer. And what UV does to us, ultraviolet, is normally the base, adjacent base to pyrimidines are separated by 3.4 angstrom, and they are, each can serve as a template for DNA polymerase. Upon UV irradiation, those pyrimidines gets cross-linked doubly, and the two bases are linked together like a Siamese twin. And another small portion, 20%, the two bases become 90 degrees to each other, called 6,4 product. And because this is more different from normal, actually repair of excision, this is a better substrate to be cleaved off. But this one, because it's quite similar to the normal base, and it's very harder, much harder to be removed by the DNA repair removing crew. And so there are 8 genetic groups that mutations cause people to oversensitive to sunlight. 7 of them are involved in this repair pathway to remove the lesion. And the disease is called xeroderma pigmentosum, and the A, B, C, D, E, F, G are all genes involved removing and resynthesizing. And the 8th gene is the variant gene that's encoding the DNA polymerase. And the role of that is because in the cell cycle of the DNA synthesis time, it's not all lesions are removed. And during synthesize time, S phase, there's no repair because it's very dangerous, you're cutting DNA to pieces. And so need a special polymerase in human to bypass 1, 2, 3, 4 steps, insertion base opposite the lesion and extend it. And Pawl has been shown the most faithful and the most efficient in cross the barrier of thymine dimer. So, and there are a lot of polymerase in cell, and they need signals to recruit those special polymerase to the replication fork. And ubiquitination is important for pCNA when cell is encounter sustained UV lesion, and the pCNA when it's ubiquitinated, monoubiquitinated, it can recruit the special polymerase called the Pawl to bypass this roadblock. And the Pawl will carry the torch, go through 4 steps of synthesis, and hand over the now fine replication fork back to the good polymerase. So we were very interested in human Pawl, and it's because the human protein is a little more difficult to get soluble enough protein, and we even had a false start. A postdoc, Santi, and a summer intern, Mark. They spent 2 or 3 months actually got the crystal and solved the structure, and turned out the structure we solved, the protein-DNA interaction wasn't exactly the native interaction because protein-protein interaction in the crystal lattice somehow distorted interaction of the important catalytic. I will show you the difference between not so perfect protein-DNA complex versus perfect DNA complex. The protein just moved very subtly, but to get the right structure, we spent another 2 years, and that's a team of a postdoc and a student, Ye Zhao and Christian Bientefel, and they spent 2 years engineering new crystal lattice so that we can see exactly when the enzyme is in the catalytic mode. And showing here is how the enzyme accommodate this thymine dimer in the active site, not making any errors. It's inserting the A opposite the lesion base. And normal polymerase just wouldn't take 2 base pair into an active site. It will close off here, and by having this, it won't be able to close. And Ye and the Christian actually went on to solve 3 more structures of the lesion with the DNA in each step of bypass, crossing this base, crossing next base, and next base. And I won't bore you with all the structures because they look quite similar. And showing here is that just how the DNA superimposed. The lesions here superimposed on normal DNA. It looked very normal. And about just 5 or 10 minutes ago, I was telling you, a good DNA is like a good spring, and the DNA-containing lesion is sort of a broken spring. And it's true it's broken if it's complex with a repair protein showing here. It That's what a broken spring looks like. Normally when you have a UV lesion and it's being repaired by repair protein will exploit this flexibility and bend the DNA. But somehow when it's complex with this lesion bypass DNA, the DNA actually was able to keep DNA straight. The reason is that if you're building a wall, you want to have the brick laying squarely on top of another. If the wall's leaning over, you can't just build up anymore. So the polymerase is somehow using very extensive protein and the DNA interactions to keep this DNA normally would bend straight for the chemistry to take place. And we liken this as a molecular splint. So the protein polymerase will keeping the DNA straight in order for DNA synthesis to occur. And at least 5 mutations that's found in the XPV patient, xeroderma variant, pigmentosum variant patient, the 5 mutations affect the sturdiness, stableness of this molecular splint. For example, arginine here supports this protein backbone that's interacting with the template. When it's mutated to serine, then it's no longer as good as bypassing this lesion. In the last couple of slides, I will turn the attention to the application of this study of this special polymerase. So Pawl is very good to help us to guarding against the lesion caused by UV, but Pawl is also play a negative role in treatment of cancer patients. Cisplatin is a very broadly used chemotherapy agent. It damages DNA by cross-linking adjacent purines. Here is UV-induced thymine dimer, have 2 covalent bond. And here is cisplatin, the platinum is here, it cross-links to adjacent guanine. And that's how it helps cell to die, kill the cancer cell, because so many of the lesions here, cell will be stopped in replication, the fast dividing cancer cell. And it turned out that the study based on cancer-treated patients shows that if you have high level of Pawl expression level, showing here, showing this curve, the surviving time is much shorter than if you have low Pawl expression level, because Pawl probably bypass cisplatin caused lesion just like UV lesion. And in stage 3 disease patient, the difference is even more severe. So if you have lower Pawl, patients respond much better to cisplatin treatment and live longer. And biochemically, other group and as well as us have shown that Pawl can bypass lesion, and the efficiency of inserting against the cisplatin is nearly 80% or 85% of inserting against normal template. It drops a little when it goes on the next base and drop much further when it's go on the 3rd and the 4th. But the first 2 are the hardest one to get by in cell. So we were looking for ways to inhibit Pawl. If you can inhibit Pawl and combine with cisplatin, platinum-based treatment, the patient will probably respond better with lower dosage and survive longer. And it was all in the talk with people in Rockville, the drug screen group, and also Roger Woodgate who were talking about it, but we serendipitously find something in the crystal structure. So we were crystallizing with pretty high concentration of incoming nucleotide, and we found there's a second binding site between the protein and the DNA away from the active site. And we were kind of bothered by this density and like how to get rid of it. And if we soak the crystal with no incoming nucleotide, we can get rid of it. So it convinced us that this is a second nucleotide binding site. And what's more interesting is that Yin and the Mark again here find that normally when you increase the substrate concentration, you see the activity goes up. But with this Pawl, when we increase after 0.1 millimolar or even 50 micromolar, the enzyme gets inhibited. And inhibition can be removed if we remove this, mutate this tryptophan, which is forming the aromatic stacking with the base of the incoming nucleotide. If we mutate this tryptophan, which is not conserved in most of the Y family polymerase or any of the Y family polymerase, when we mutate this to alanine, now the enzyme lost some activity, we think because the folding and the interaction with DNA is affected, but it's no longer being inhibited. So this may be a good target for finding new drugs to inhibit POL-A and the use for cancer therapy. So I'll conclude my talk here. So I showed you about DNA lesion, which can be caused by external effect or environment agent, or just because polymerase make an error. And those lesions leads to increased flexibility in DNA. And it's used by both repair enzyme assisted by ATP for repair, or fortified by special polymerase in order to carry out translesion DNA synthesis. And in both processes, the outcome is prevent the mutation and reduce the cancer incidence. And the side effect of POL-A may be leading us to think about developing drugs for inhibition and helping effectiveness of cancer treatment. So I've acknowledged the people, I talked about the work, and here's the current group. And the group looks a little larger because we have picture taken together with Marty's group, and it's a summertime, we have expanded lab. And a lot of people's work I'm not mentioning here because they're still in the working with some larger complex. And I mentioned my former people and also former postdoc students and also collaborators. And for POL-A, we collaborate with Fumio Hanaoka in Japan. And there are a lot of wonderful collaborators, many of them you recognize from NIH. And we are supported by NIH intramural funding. And then lastly, but not the least, I want to acknowledge Laboratory of Molecular Biology at NIDDK, and many of them are here. And I feel sort of highly pressured and very nervous because I'm just representing our group, because all of them, every one of them have a wonderful story to tell, and it's a great group to belong to, and the senior member really inspires me by doing the science the way they do, and in action they show that what's important, and it's just great fun to be a member of this family. And I also belong to other sort of group in NIH. ITAP has been very supportive, and I have a lot of positive, interesting interactions with members in the ITAP. And also the Society of Chinese Bioscientists in America. In NIH, we have sporadic interactions and very, very supportive and forming a lot of conversations and hopefully productive collaboration in the future. Thank you all.
Questions?
Here.
Yeah, could you?
Great talk. So my question is, when you form nucleosome in the cells, so how does nucleosome interact with the MutS? And is there any difference with the methylinic DNA?
So you mean the lesion search?
Yes.
Right. So it cannot be sort of sliding on DNA forever. It's only sliding on very short distance. It has to be it's a combined 3D, 2D search. So dissociation is important, and then we'll hit another spot.
Okay, thank you.
Yeah, it's usually combined 1D and 3D movement inside the nucleus.
Hi. Have the structures given you any hint as to how the complex can tell the old DNA strand from the new DNA strand?
Right. In bacteria, in E. coli, it's not all of them. So in some bacteria, it's clear because the template in E. coli is methylated and the daughter strand is not methylated. So the nucleus will be only cutting the unmethylated strand at a hemimethylated specific sequence. And in other bacteria, it's really not clear. But in human, the general consensus is that because human sort of polymerase doesn't move very fast, so there's always the daughter strand always have a 3' OH, the end of the primer strand, to be recognized, versus template strand is totally continuous. But this is still, jury is still out. Because we haven't found the protein, the nucleus that cleaving the daughter strand specifically, and there's also sort of more recent work that MutL can cut daughter strand. If it's helped by pcNA and the MutS, you have like a three-way interaction. Then you set up the scene right because the pcNA the loading clamp will be only loaded in certain way relative to the primer strand. So it needs helpers to differentiate the two strands.
So I'm afraid this is going to be a rather arcane question, but it comes from my interest in heteroduplex DNA and recognizing that a CC mismatch actually is one of the most destabilizing of the DNA duplex. Yet, it is the mismatch that is not recognized by MutS.
Not recognized well by MutS. You know why?
No, I don't know why, and that's why I'm asking you.
So the answer is the two sides of the coin. Polymerase doesn't make CC mismatch very much because CC is so unstable. Right. So the polymerase doesn't make it very much. Right. It's the same side. It's two sides of the coin because it doesn't make very much, very few. It's only human made in two.
But in terms of the molecular mechanism and in terms of what you said in terms of probing the DNA structure for flexibility, I would have expected it to be the most flexible.
It's somewhat different. No, it's different. Okay. Yeah, so normally the base will be still stacked in, just stack weaker, so sort of like doing this.
Right. Okay.
But the CC is so bad that most likely it's in vitro study, they kind of pushed out, and adjacent base becomes stacked. Okay. And that become more stable. And because the base pushed out are not very well recognized by MutS, it's recognized by other pathways, like base excision repair.
Oh, wait, that was beautiful. So with the Pawl Eta story, where you show the splint straightening the DNA, but before Pawl Eta gets there, the DNA is bent. Do you have any idea how the DNA becomes straight enough to load Pawl Eta? Well,
the DNA actually is not always bent. What the increased flexibility means that it isn't, normally the DNA is this. If it's have a lesion, it does this. It's just have residence time in very little in every state. But it could be straight, it's just very little. So the polymerase just captured it, went straight, and it kept it straight. And that's most of the saying, like we say, induced to fit. And probably 95% of the time, it's not induced to fit. The conformation is already there, it's just not very stable. But by interaction, it gets trapped.
Before I thank Wei, I wanted to make a couple of announcements while I have this captive audience. The first is that the Wednesday afternoon lecture next week is on Thursday, so change your calendars accordingly. Secondly, we had another interesting event here at NIH, which was sponsored by the journal Science, a webinar on applying new imaging techniques to your research. This is so-called super resolution imaging, not the resolution of X-ray crystallography, but pretty good. And NIH today actually recorded a discussion with Harry Schroff, who is a new tenure-track investigator here, Sriram Subramaniam, and Claire Waterman. And that webinar will take place officially on Wednesday, February 29th at noon. So if you're interested in checking in, I think you might find that an interesting event. So I just wanted to say that in discussing this lecture before Wei got on the podium, she told me she felt the burden of representing the laboratory of molecular biology, which, as you all know, is one of the gems at the NIH, and the institute that she's in, NIDDK, and women scientists and Chinese American scientists, and she was worried that she would acquit herself in the best possible way. And I can assure you, Wei, you've done a magnificent job. Thank you very much for that lecture.